Hi, good morning. My name is Mary Park. It is week 2, lab 2, and it's on aseptic technique and culturing microbes. So, culturing microbes in a broth. In this experiment, I will be inoculating nutrients broths with an active microbe samples using aseptic technique. The broth will be incubated and examined for signs of any microbial growth. So what I have done, so the experiment has been completed already prior, but now I'm actually going back to actually how to do the experiment. So what I have here right now are your nutrients broth of what you would need, and right here is your E. coli culture as well as your epidermidis and your S. cerevisiae culture. And right here is the alcohol that will be needed, and the yeast to mix it, and your candle for doing aseptic technique, as well as your candle lighter, and what you need to make sure that we practice safely, as well as your gloves, your gowns, your mask. And I also too have a what you would put over your clothes. Okay, so these is what you would need. These are the materials that is needed to actually do your culturing microbes in a broth. So I would go ahead and talk about it. So, sorry. So you would need to make sure that your E. coli right here is, this would been your nutrient nutrient broth, and you would want to go ahead and make sure that you would pipette 0.25, which is like a drop, into this actual culture right here, and you would want to mix it. You would want to mix it, and then you would want to mix it and mix it right here to create an actual culture for the future for you to grab. And then same thing as for your S. epidermidis, you would pipette 0.25 broth from the nutrient tube into the culture vial, and you would go ahead and mix it again. And then same thing with your S. cerevisiae, same thing you would do. But for the yeast part, you obviously would first want to mix it for half a teaspoon and add 1/4 cup of water in order to actually get your culture so that you can actually put it into the broth. Then eventually, before all doing all this, you want to make sure you do practice aseptic technique. And by doing that is definitely by, inoculating it with just making sure that you hold on to the cap, and you would go ahead and just make sure you do it while. And same thing goes for your other 2 tubes as well. Let's see. So, and then you definitely want to make sure that you would incubate it for a good about 48 hours. And so when you do that, you'll develop cultures will either appear turbid, which is like cloudy, and if culture shows no sign of growth, then after 48 hours, then you want to go ahead and incubate it for an additional 24 hours. So because I've already had done the actual experiment already ahead of time, I just want to show you exactly what it looked at that time when I had completed the actual experiment. I wanted to show you how it looked like after the 48 hours, and I will definitely show you the pictures of how it would look. So This is the E. coli culture, and with the E. coli culture, obviously you see some turbid. So that definitely shows that there has been growth. The second picture is my S. epidermidis, and you can only see from the bottom that there is culture as well, that there is growth, sorry, that there is growth. In my photo number 3 is my Saccharomyces cerevisiae broth, and also too you can see that there has been turbid, so there definitely has been growth. And again, here is my culture results, but I'll go ahead and see that to the next video. Thank you.